Thank you very much. Thanks, Amber and Monica, for inviting me to this amazing pre-meeting, and really learning a lot, and really hopeful that I can talk about some of the work that we've been doing in Boston Children's Hospital, and very complementary to what Dr. Gray has just talked about in the gene therapy space. We're coming from a slightly different angle. We are focusing on a cousin of SLC6A1, if you will. It's SSADH deficiency. In the first few slides, I probably will tell you about what is the story behind this, and what is our approach to develop a relevant gene therapy for this GABA-related disorder. And you might be curious, what is Galibra? Actually, it's a initiative. It's actually a company that we just founded earlier this year. The focus really is a subset of this GABA-related disorder. And the Galibra, so me and my mentor, Dr. Rottenberg, sitting at the back here, is the co-founders of this company. And really, as you might know that Galibra, so we are using gene therapy to try to balance GABA. And Libra is an astrology sign of balance, so that's why Galibra. So the objective today I would like to talk to you about is that we will review some of those scientific rationale of gene therapy for GABA-related disorder. And we will talk about some of the preclinical studies that we have been working on in this GABA catabolic disorder called SSADH deficiency, and some of those knowledge that we gain from knowing this disorder and how well we can develop a gene therapy may be scalable and transferable for other related disorder, including SLC6A1. So just an overview, as a scientific audience, I don't need to go through this to detail, but I think the take-home message that there are many genes that are actually relevant for a life cycle of GABA, and many of those are actually related to developmental disorder, given the profound function of GABA in the brain development and inhibition. And so for SSADH, it's a rare disorder. And it's Boston Children's Hospital is the major global site for natural history study for SSADH. And that's why we have the very strong clinical background for this disorder. And we know very well about what is the GABA doing in the brain, what is the synthesis, what is the recycling, what is the degradation pathways. So given the maturation of the technology these days for gene therapy, really precision medicine, so we think that gene therapy might be a amenable and really a disease-relevant treatment for this disorder. SSADH is an autosomal recessive disorder, so it's slightly different from SLC6A1. In SSADH, both genes of ALDH5A1 is mutated, and there's a total loss of function of the enzyme, which results in accumulation of GABA because of the chemical equilibrium, and also a byproduct called GHB, gamma hydroxybutyrate. So GABA and GHB are accumulated in the brain, in the developing brain. It's a neurochemical, biochemical hallmarks of this disorder. And there are only under 500 documented cases as of this year, so it's an ultra-rare disorder. But we think that this related disorder, including SLC6A1, maybe we're thinking in the order of tens of thousands of patients out there that might be amenable for this gene therapy, because we know precisely where the genes are having a problem, and then we can use this gene-targeted approach for a cure, potentially a cure. And so, what we have done preclinically is that we want to ask two basic questions, and is that one is that whether this disorder, the phenotype that is exhibited in patients or in mouse, whether they are reversible. When we treat them when they already develop the symptoms, can we still see the reversibility of the phenotype? The second is what is the threshold for this gene replacement? We know that we need to put the gene back, a functioning gene back. The question is how much and what is the threshold for that? So using a mouse model, we think that will be a very good way to try to understand what is the range and what we expect when we can set R. And knowing those information in parallel, we can also design a vector. Dr. Gray talked about very nicely about using different polymerase strategy and different endpoint. We can look at the mouse, and also some of the finding that he found that might be relevant, for example, heterozygous and homozygous comparison. So we really is really going in kind of parallel, but not focusing on SLC at that moment, and we have been doing for the SSADH. But we think some of these underlying biology might be transferable. So this is just a cartoon showing what we think about SSADH. Why the patients they have seizures when you have accumulation of GABA? That could be applicable for SLC. And we think that it's because the brain is plastic. They are developed in a way such that the GABA receptor is actually downregulated, and it's actually proven clinically. But when it comes to gene therapy, that we want to be assured that this reversal of the downregulation of GABA receptor can be reversed, and that reversibility might be critical for a successful gene therapy. The other factor that we think about is that SSADH expression is not ubiquitous, although it is widely expressed. But you do see that from this in situ hybridization, some of the brain regions are expressing a little bit more of SSADH than the others. So panel B is a single-cell RNA-seq panel that you can see from this Manhattan plot, if you will. There are cell types, including interneuron, that actually express a little bit more of SSADH compared to the others. So the bottom line is that we want to put the gene back to the relevant cell types, the correct gene, and then correct cell types in a correct dose. And how are we going to mimic that? And so we make this mouse model. It's a lockstop mouse model. Simply put it is we basically put a switch to switch off the gene. So the animal will develop in a way where there's an absence of SSADH. Very much similar to a patient situation. It's a homozygous situation because the patients do have a homozygous mutation on that. But because this is a Cre-dependent strategy, so we can introduce this Cre recombinase, which normally does not exist in the brain, in the mouse brain. And we can introduce this Cre in a cell-specific, time-specific manner. And we can switch on the gene and then to see how the animal can react to it. Does the phenotype go away? Does it cure at all? So this is a kind of basic characterization of the mouse with the help of a very talented research assistant, Gabrielle, in the lab. So panel A shows you the Western blot showing the amount of SSADH, just a proof of concept to show that in the heterozygous animal, there is a 50% of the reduction or 50% of the SSADH expression compared to wild type. In homozygous, there's none. Virtually, there's no SSADH expression. So showing that this lock-stop strategy is very with a very high fidelity in terms of the restriction of the expression. And at the same time, we do see from these mice at P16, there is already like over 500, I think it's like almost 1,000-fold of GHB, which is one of those biochemical hallmark of SSADH, can be found in these homozygous mutant mice. So in terms of survival, we also see that all the mice actually develop seizures starting from 2 weeks, and by 3 weeks, they all expire. So this gives us a very good model in terms of survival, in terms of some of the relevant biomarkers, and also the underlying biology seems to be what we expected. So now goes to using AAV, so which will be a vector that we will eventually use, but it's a proof of concept. We just carry a Cre gene under a very strong promoter, and we just ask the question whether this animal can be rescued at all. So we know that we need to target the brain, but the way that we do it in a very young animal is by systemic injection. So by doing intraperitoneal injection, IP injection, and the virus will pass through the blood-brain barrier and infect into the brain. So you can see in the panel B that the curve actually, there are two curves. Red one is actually injected with the virus, but somehow it did not work, and half of them actually still die. But the other half actually has a very good survival. So I'll show you some video here on the C panel before treatment. I can show you that. Yeah, so you can see that animal before treatment is wobbly, they have seizures, they are underweight, and they will die at 3 weeks. And we treat the animal at 2 weeks. This is only giving them 1 week of runway for rescue. And we actually can see that this is on the right, a pair of animal, one is wild type, the other is a treated homozygous at 100 days. So you can actually see that the lifespan can extend by at least 5 times as well more. So this is a very promising, and as a proof of concept, it is great because we see that the animal is already showing the symptoms and is totally reversible. And then we want to ask the question, why some animal will work, why not some of them? And the question simply is that actually the virus does not go to the right place. So panel A shows you the Western blot, but now showing you on the last 2 lanes in here, one has absence of SSADH. Those are the one that injected with virus but still died. And it turns out that the virus just does not go to the brain, and for some reason, because we injected IP. And but for those that survive in the 3rd lane here, it's actually have a very good expression. It's actually over 50% in average. So that is a level that is, we know that it is tolerable because all the heterozygous animals are asymptomatic, as same as the parents. They are carriers. They only have 50%, but we know that with that level, it's tolerable. So that actually will be a good point for SLC6A1. It's quite different because SLC6A1's haploinsufficiency, we know it's a loss of function. 50% probably it's not. It's actually pathogenic. So in terms of the restoration threshold, we might need to set it higher. But that's actually something that we can totally, use in the mouse model to mimic that situation. And on the panel B, just to show that this expression of SSADH or restoration actually is correlating with the amount of GHB, this biomarker in the blood. But very interestingly, even in an animal that is rescued, we actually see that the amount of the GHB actually is still higher than the wild type glutamate, so suggesting that the level of GHB, even though it's a little bit higher, but it's actually tolerable. So mouse proof of concept is great, but no patients have a switch for us to switch it on. So we actually still need to make the vector. So this is the strategy that we that we use, is using a native promoter because as Dr. Gray has talked about, that promoter might play a role in how to reconstitute that expression pattern of expression that is mimicking that endogenous ones. So we take about 1.7 to 1.8 of the upstream of the transcriptional starts of the ALD5A1 gene that encode SSADH, and then we put it into the vector and to see what happened, and injected this virus using the same capsid that can pass the blood-brain barrier and deliver into the mouse. And so this is behavior showing you on panel A is that before treatment, these mice are hyperactive, they are asocial, and they will die at 3 weeks. But after injection of this vector, without doing the switch, we actually see that the animal is actually behaviorally there's a reversal on that, and they become social, and they are not hyperactive, and they also can live. In terms of the survival, we see that the animals, actually 50% of them can live to 100 days. So that's approximately 5 times of the lifespan of an untreated animal. We do see some animal that seems initially having a response, so as shown in panel B in those green curves, but then they will still expire between the age of 30 to 60. So that's the next question. It seems that there are some animals that initially show a response, but they still die. And the question is why? So when we looked at the brain and also their internal organs, it seems that it's very interesting because there seems to be a correlation between the expression of SSADH to their the survival and the outcome. So the green curves on the green bars on the left is that showing you all the full rescue animal. Those made it to 100 days, and you can actually see that the expression in the brain and also in the heart is actually highest. And compared to those partial rescue that is represented in the yellow bars that are lower. And correspondingly, for those that are not rescued but injected the virus, actually has very low level of SSADH. So the expression of SSADH in the cortex and the heart seems to be correlating to the outcome of this disorder. So we are hopeful now that we know where to target and what is the level that we need to achieve. So in summary of all the work that we have done so far for preclinical study for SSADH, we have gone through the proof of concept stage, and we know that there's a reversibility of the phenotypes. We also know there is a expression threshold that we need to cross in order to have these therapeutic effects, and we also know the brain targets that is relevant. And we also know that using a full-length native promoter strategy, that seems to be very good in terms of driving the expression of SSADH, at least in the mouse. So that comes to like what we are talking about more broadly. What we learn from SSADH, we think is totally transferable to other GABA-related disorders. And you look at the timeline of what we have done, the work that I've shown you so far in the last 10 slides or so, actually started just around the pandemic time and with all the slowdown and so on. So we think that because we have all the structure, we have all the support from clinical side and also the basic science, what we know so much about the GABA and their metabolic disorder. We can already gone to this stage from getting the seed funding from SSADH Association to where we show this efficacy data using the vector, which is a potential candidate for gene therapy, is within 3 years. So last year, I reached out to AES, I reached out to Amber, and then we also start thinking about what is the next indication that we can use so we hopefully we have done it for one disease, we can do it the second one, and maybe this time we can even be faster. So our team basically we started very small. So it's my mentor, myself, and Amber for this Galibra team. We have good advisors in the clinical side and also the AAV expert, Dr. Pearl and Dr. Gao. And just show you a little bit of our logo, which is my design. And I don't know how many Easter eggs you can actually get it from this logo. I am very proud of myself and making this because you can actually see that there is a Libra sign. And if you don't see, I can tell you later. And there is a synaptic vesicle releasing GABA, I think. And there's a balance, right? So I'm very proud of this design that I come up. And so just to think about a little bit of the due diligence. So we're thinking about, okay, the next one is SLC 681. So would that fit with what we have done so far? So the answer seems to be yes. This showing you some of the molecular biology behind the promoter. So we can use the same strategy and we can make a vector. And we know also from a brain study where the expression of SLC 681 is. So but specific for SLC 681, as I said, it's not the same as SSADH. So the first question is, do we have a mouse model that we can mimic that situation? The answer actually is yes. So in the afternoon, we will hear from Jennifer from Jackson Lab. So they actually developed a lockstop mouse model, which I think many of you have known. This is still in the early characterization stage, but we think that could be a very useful tool for us to understand the same way as SSADH how restoring a gene can help with the reversal of the phenotype. And then to be more specific for SLC 681 is that SLC 681 is autosomal dominant. So one one copy of the gene is actually functioning and the other one is not. So given this haploinsufficiency situation, will that addressing the haploinsufficiency enough for reversal or producing this therapeutic efficacy? That's something that we think about, and but we also think about that maybe because there's another copy that is not functioning, and that's very likely to be a variant, truncation, and so on. So will that also have some functional impact on the wild type protein? If we replace a good gene or boost up the amount of the good gene, is there any dominant negative effect, if you will, that might affect the good protein? So these are the things that we can empirically test in, if not in a mouse model, we can also test it in a in vitro setting. So Dr. Christmas Squirrel will talk about in the afternoon, I believe, that he has a very nice GABA sniffer, just a little bit of the teaser at this time. And so that we are thinking that could be a very useful tool to understand the functioning aspects of this gene therapy. So at the end, I would like to acknowledge, thank my mentor, Dr. Rottenberg, sitting at the back, is always very supportive and bringing me from a very basic science perspective to a clinical stage is really something I've never dreamed of. But thank to Dr. Rottenberg for bringing me into this very interesting journey and very unexpected, but very fulfilling and very thankful for that. And also a lot of people in the lab is very helpful with different types of experiments and their specialties. And we also have a lot of institutional support from other labs, from Team Yu's lab, from Mustafa Sahin on the AS antisense oligos and also the iPSC's aspects. And of course, we have the clinical side. Dr. Pearl is a beacon for us at the beginning because he is in this space for a very long time. And he actually connected us to the SSADH Association for funding initially. And we also have Dr. Guangping Gao as our experts in the AAV space and the capsid and the delivery approach. So really benefit from really a talented and specialty team, and also the funding from SSADH Association and NIH. But last but not least, I want to thank Amber for her approval, if you will, of our work and really interest in our work and really going to the next stage and thinking about beyond SSADH, maybe SLC6A1, some of the strategy that we learned, some of the knowledge that we have we've gained in the past few years is totally doable and maybe transferable for this disorder. And also want to thank Steve Gray and for our very fruitful discussion, and some of those previous work that he's done is really helpful for us to navigate in this situation, and Jennifer and Chris for the collaboration potentially. So thank you very much for your attention.